Welcome to Pearson Bio Academy. If you are new to my channel, this is Fazna Imran, and this is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Currently, we are focusing on Pearson Edexcel International Advanced Level, paper reference WBI 1401, Unit 4, Energy, Environment, Microbiology and Immunology. And under photosynthesis, we are tackling questions from 2020 to 2024. For better understanding, I've divided the topic of photosynthesis into subcategories. They are structure of chloroplast, absorption and action spectrum, chromatography, photosynthetic reactions, and finally photosynthesis products. And today we'll be focusing on absorption and action spectra. First, let's look at the absorption spectrum. In an absorption spectrum, we are observing how much light is absorbed by different photosynthetic pigments across various wavelengths. And this helps us understand the specific wavelengths that chlorophyll and other pigments absorb most effectively. And also the absorption spectrum shows that different pigments absorb light at different wavelengths. Let's start by looking at the absorption of light by different pigments in plants like chlorophyll A, chlorophyll B, carotenoids, and by whole chloroplast. Chlorophyll A absorbs light strongly in the blue and red parts of the light spectrum, so it means they capture a lot of energy from these colors. And you can see the wavelengths they cover. And chlorophyll B also absorbs blue and red, but a bit differently. In the blue region, it absorbs slightly more than chlorophyll A, while in the red, it absorbs a bit less. The red absorption of chlorophyll B also shifts slightly towards the orange area. Now, carotenoids are a bit different. They primarily absorb light broadly in the blue region, much more than either chlorophyll A or chlorophyll B. However, they do not absorb in the red region at all. By covering a broad part of the blue spectrum, carotenoids help capture light that chlorophyll A, B might miss. And when we look at all pigments together in plants, we get a broad absorption of spectrum covering a wide range of light. So this combined spectrum ensures there is no point where their absorption drops to zero. So each pigment absorbs light in its own way, but together they cover almost the entire light spectrum that plants receives, effectively in the blue and red regions, maximizing the light energy available for photosynthesis. Now let's talk about the action spectrum. The name itself suggests action, and in this case it refers to the action of photosynthesis. In an action spectrum, we are looking at the rate of photosynthesis. It means how much photosynthesis happens over a certain period of time at different wavelengths of light. So the main question here is, at which wavelengths is the rate of photosynthesis highest or lowest? So by studying the action spectrum, we can see which colors or wavelengths of light are most effective for photosynthesis. If we look at the action spectrum, we can see that it's never zero at any point. However, it does show maximum photosynthesis rates in the blue and red regions. When we compare the action spectrum with the absorption spectrum, we notice they both align closely. So this means that where absorption is high, the rate of photosynthesis is also high as the pigments are absorbing the most useful light. So both spectra show that the blue and red wavelengths are the most effective for photosynthesis, and importantly, there is no point in either spectrum where the values drop to zero. So it means there is some absorption and some photosynthetic activity across the entire spectrum. So this allows plants to make efficient use of nearly all the light that reaches them. Why do most plants appear green? Well, most plants look green because of these chlorophylls. The chlorophylls absorbs light from the blue and red parts of the light spectrum but doesn't absorb much green light. Since green light isn't absorbed, it's reflected back to our eyes, making the leaves look green. And this concept applies to all colored objects. For example, if you see a pink object, it means that object is absorbing all other colors except pink, which is reflected back. So this idea might seem basic, but it's actually a key concept in understanding how we see colors. Surprisingly, this idea had been checked in your past paper as well. Seaweeds often come up in questions about light absorption and the action spectrum. Even though they look like plants, they are actually a type of algae called sea algae. Getting familiar with names Corallina officinalis, Sargassum linifolinus, and Alva could be helpful as they may appear in questions. So knowing these names can give you a quick advantage when you see them. Let's look at the first question from the January 2020 paper. It involves the composition of three seaweeds, Alva fasciata, Sargassum linifolium, and C. officinalis, collected from a beach in Egypt. The graph shows the chloroplast pigments extracted from these three seaweeds where the concentration of these pigments, chlorophyll A, chlorophyll B, and carotenoids are given for each seaweed. Before we analyze the graph, let's quickly check the question. We are also provided with the absorption spectrum for these three pigments. The absorption spectrum shows the specific wavelengths of light absorbed by each pigment. And the question asks which row of the table shows the color of each of these seaweeds based on their pigment composition. Let's begin by analyzing the absorption spectra of the pigments involved. First, we'll look at the absorption of chlorophyll A. As we can see, chlorophyll A absorbs light most effectively in the blue and red regions of the spectrum. The absorption in the blue region is relatively high compared to the red, but it still absorbs both wavelengths strongly. Next, if we examine chlorophyll B, we can see that it also absorbs in the blue and red regions. But chlorophyll B absorbs more blue light than red light. Next, let's focus on carotenoids. Carotenoids absorb primarily in the blue and blue-green regions of the light spectrum. They absorb a significant amount of the light in these regions, but are relatively weak at absorbing light in the red or orange areas of the spectrum. Now let's summarize the pigment composition and colors of the three seaweeds. Alva has a high concentration of chlorophyll A, a moderate amount of chlorophyll B, and very low level of carotenoids. So the abundance of chlorophylls allows it absorb the blue and red light, reflecting green light, which gives its characteristic green color. Sargassum linifolium contains less chlorophyll A and B compared to alba fasciata and moderate carotenoids. With lower levels of chlorophyll, it absorbs less blue and red light. The carotenoids, which absorb in the blue-green region, reflect yellow, orange, and red light, resulting in a brown appearance. Say officinalis has abundant chlorophyll A but very low chlorophyll B and moderate carotenoids. The high chlorophyll A absorbs blue and red light, while the low chlorophyll B means it absorbs less red light. The carotenoids absorb blue-green light, so reflecting more red light, giving the seaweed a reddish appearance. So now the question asks you which row of the table shows the color of each of these seaweeds, and your answer is green, brown, red. The next question is a straightforward one from the October 2020 paper. The statement says an absorption spectrum shows how much light is absorbed by chloroplasts at different wavelengths of light. The table shows the color of light at 4 wavelengths. 460 nanometer is blue, 520 nanometer is green, 600 nanometer is yellow, and finally 680 nanometer is red. Now here is the question, which wavelength of light is absorbed the least by chloroplasts? This is simple if you understand the basics of color theory. Since chloroplasts appear green, which gives green color to the leaves, they reflect green light more and absorb it less. So the answer here would be the green wavelength, which is 520 nanometers. The next question, also very direct question. You have to write the meaning of this term action spectrum. So the action spectrum means the rate of photosynthesis at different wavelengths of light. The next question from January 2022 paper. Here is the statement. It reads, thylakoid membranes contain chloroplast pigments and molecules of ATP synthase. We know that. These membranes are the site of the light-dependent reactions. The graph shows the absorption spectrums of 2 chloroplast pigments from one plant. Here we have a graph. The x-axis shows the wavelength of light and the y-axis shows the percentage of light absorbed. In this graph, we see the absorption spectrum for 2 different pigments plotted together. As I always say, when you have a graph, it's better to go to the questions first before diving into the details. The question here asks you to draw the action spectrum based on this plot on the axis below. So here, how we will approach it. First, we will plot the points by marking the top contour of the 2 graphs, essentially adding up the absorption of each pigment. We will mark these points along the plot. Then, using these points, we can roughly draw the absorption spectrum, which will give us a good approximation of the action spectrum. So now let's draw the action spectrum. Remember, we are working with an absorption spectrum on the given graph, but we can approximate an action spectrum. As we've learned, the action and absorption spectra are very close to each other, so we can use this to guide our drawing. And also you can draw the graph like this, making sure that the left peak is always higher than the right one. And according to the marking scheme, the line we draw should roughly follow the shape of the absorption spectrum lines and should not drop to zero. Also, if there is a left-hand peak around 460 nanometer, it should be higher than the right-hand peak. And this will give a good approximation of the action spectrum based on the absorption spectrum. And next question, you have to calculate how many times greater the percentage of light absorbed by chlorophyll B is than that absorbed by chlorophyll A at their maximum absorption. So for this question, we will need to analyze the graph to find the maximum light absorption percentages for each pigments. By examining the graph, we see that chlorophyll B has a maximum absorption of 85%, while chlorophyll A has a maximum of 69%. To find out how many times greater the percentage of light absorbed by chlorophyll B is than that of chlorophyll A, we will divide the absorption of chlorophyll B by chlorophyll A, and it's 85 divided by 69. So the final answer is the percentage of light absorbed by chlorophyll B is 1.23 times greater than that of chlorophyll A there at the respective maximum absorption point. So the next question is from the January 2023 paper. Here you are given a graph showing the absorption spectrum of photosynthetic pigments in both a plant and a bacterium. So this is the graph and you can see both absorption spectra. First, let's go through the question quickly. The questions ask for two conclusions that can be made about the differences in absorption spectra shown in these two graphs. And we just need to draw two conclusions. So looking at the graphs carefully, one key observation is that the bacterial pigments compared to the plant pigments are absorbing a wider range of wavelengths. So you can see the bacterial pigments absorbs light around from 320 to 900 nanometers, while the plant pigments absorb in a narrower range from 330 to 760 nanometer around. So the plant pigments have more limited absorption range, and we know that after 760 nanometers they can't effectively absorb light. And next, if you look at the maximum absorption, the bacterial pigments absorb maximally at 380 and 880 nanometers, while the plant pigments absorb maximally at 440 nanometers. Overall, you can say that the bacterial pigments absorb more light at most wavelengths compared to the plant pigments. So in this question, you are asked to describe two conclusions based on the absorption spectra of the both pigments. Although I have given you three conclusions, two of these are enough to score the 2 marks. Let me go through them. The first conclusion, bacterial pigments absorb light at a wide range of wavelength than the plant pigments. So if we look closely, bacterial pigments can absorb the light from 320 to 900 nanometers, whereas plant pigments can only absorb light in the range of 330 to 760 nanometers. And the second conclusion regarding the maximum absorption, bacterial pigments absorb the most light at 380 and 880 nanometers, while plant pigments absorb most light at 440 nanometers. And the third conclusion, overall the bacterial pigments absorb more light across most wavelengths compared to plant pigments. The next question is from the 2024 June paper, and it's about seaweeds and light absorption. The diagram shows the position of three different types of seaweeds on rocks covered in water, and it also shows how far red, green and blue light can penetrate into the water. Let me explain the concept first. First, let's talk about why the position of each seaweed is important. The depth at which each seaweed is located determines which types of light can reach it. Red, green and blue light don't penetrate water equally. Red light penetrates the least and is absorbed quickly by water, so only seaweeds at shallow depths can absorb it. Green light a bit deeper but not as far as blue light. The blue light with the shortest wavelength, they can penetrate the deepest into the water. And if you look the green seaweed, it is located at the depth where red light can still reach it. Green seaweed only absorbs red and blue light, but it reflects green light, which is why it doesn't need to absorb green light. So for the green seaweed, it can absorb red and blue light while reflecting green light. So this position is beneficial because it has access to both red and blue light for photosynthesis. And next, the brown seaweed. It's located a little deeper where red light cannot reach. However, green and blue light can still penetrate to the brown seaweed. So brown seaweed absorbs green and blue light but does not absorb red light. This position allows the brown seaweed to make the most of the light available to it at that depth. And finally, the red seaweed is located at the deepest point where the red light does not penetrate at all. These seaweeds absorb green and blue light. So blue light is the only one that reaches this depth. So the red seaweed is adapted to absorb it. The position is at the deepest point is beneficial for the red seaweed because it can absorb the blue light that is available there. And here the question is, which row of the table shows the light that green seaweed can absorb? So green light, the green seaweed will reflect green light, so they cannot absorb. Red light, they can absorb. Therefore, your answer is C. The next part of the question is about the position of the brown and red seaweeds on the submerged rock. So if you look at the diagram, brown seaweed is positioned at a mid-depth where red light can't reach, but green and blue light still penetrate. So it absorbs green and blue light without competing for red light. And red seaweed is positioned deeper where the only blue light can reach, and it absorbs blue light and doesn't compete for green or red light. So this separation in position helps reduce competition between the seaweeds. So the brown seaweed absorbs green and blue light at a shallower depth, while the red seaweed absorbs blue light at a deeper depth. So this way, both can maximize their energy intake without interfering with each other. And you can give your answer like this. The positions of them can be attributed to their maximal light absorption, and this separation allows each type of seaweed to reduce direct competition for light. particularly within the green and blue wavelengths, ensuring that both can optimize their energy intake without interfering with each other. I hope this video was clear and informative. If you haven't already subscribed to Pearson Bio Academy, please subscribe to stay updated with all our future content. Don't forget to hit the like button if you found this helpful. Thank you for watching and stay tuned for more videos coming soon.